Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the 8th lecture and today we'll discuss about confocal microscopy. So in the last class, we discussed about types of illumination and we discussed about wide field illumination, confocal illumination, turf illumination, light sheet imaging, two photon imaging and high-low illumination. So we briefly touched upon confocal, but we didn't go into the details of confocal microscopy. But as you can see here on this slide, in the wide field illumination, the entire cell is illuminated, whereas you just image this focal plane. Whereas in confocal illumination, a cone of light illuminates the cell, and this particular point is getting observed by the objective lens. And why these different types of illuminations are required? Just to avoid the out of focus light and just to avoid unnecessary photo bleaching and photo damage, because with the objective lens you can image only one particular focal plane, so there is no point of illuminating the entire cell and observing only this small portion of the cell. Instead, you just illuminate only that section which you are able to image. Okay, so that's why these different modes of illumination strategies were developed to avoid the out of focus light as well as to avoid the unnecessary photo bleaching and photo damage to the cell. So in confocal, as you can see here, a cone of light illuminates the sample, and this particular the center of the cone is being observed by the objective lens. So we'll look into the details that how the confocal microscopy works. So here you can see the difference in the image quality between a wide field illumination and confocal illumination. So this is a wide field image and this is a confocal image. Now what you can see here, which image is better? So obviously we will tell that this confocal image is better than the wide field image, because the details are more clear in this image compared to this wide field image. So why this wide field image is, it looks like little blurry compared to the confocal image? So because in the wide field microscope, it collects the out of focus light which blurs the image. We will shortly understand what is out of focus light, but here because of the out of focus light, it blurs the image, whereas in confocal microscope, the out of focus light is removed, and because of that you can see the structures more precisely in the image. So here are two more images to compare the image quality between the confocal and the wide field. So the left side image represents the wide field image and the right side image represents the confocal image. So the question is, if only the confocal microscope can produce this kind of crisp image, then why one should use the wide field microscope? Okay, so the answer is that for 90% of the applications, the wide field microscopy serves the purpose, but only for some specific applications which we'll discuss later, the confocal microscopy is used. And again, this technology provides better resolved images, but simultaneously it also has several disadvantages. So you have to decide whether you really need to go for the confocal imaging for this particular sample, or you can do your work with a wide field imaging. Okay. So let us see how this confocal microscope removes the out of focus light. Okay, so in this diagram, you can understand that concept. So one unique feature of the confocal microscope is the presence of a pinhole. Okay, and what is a pinhole? So you see, this is the optics of the microscope setup, and this is the optics in the emission light path. So let's say this is your slide, this is your sample, and this is the focal plane. You can see this dotted line, so that is your focal plane. So see, if your object is present exactly at the focal plane, then the rays of light will emit like this, and you can see there is a slit here which is known as the pinhole, Okay, so pinhole allows this light to pass through the pinhole. Okay, so this light will pass through the pinhole. But what happens if your object, if the object is slightly above or below the focal plane? So let's say this is the object which is slightly above the focal plane. Okay, so the emitted light will block here. It cannot pass through the pinhole and it cannot reach to the detector. If the object is below the focal plane like this, then you see the emitted light again will stop here and it cannot pass through the pinhole and it cannot reach to the detector. So only the light rays coming from the object at the focal plane, they can pass through the pinhole and they can reach to the detector. So this is how the confocal microscope removes the out of focus fluorescence light. So in confocal microscopy, what happens generally in the wide field microscope, you collect all the light which is coming from the molecules above and below your focal plane. Okay, whereas in confocal microscope, you are eliminating the light coming from the molecules, fluorescent molecules, which are above or below your focal plane, and you only allow that light to pass through the pinhole that which is coming from the focal plane. Okay, so this is how the confocal microscope avoids out of focus light to reach to the detector. Let us see it in little using a different schematic. So here also you can see the comparison between wide field and confocal microscope. So see, this is the optical configuration of the wide field microscope. Here is your sample, here is your objective lens, this is a dichroic mirror, and the emitted light reaches to your detector. And here the camera is used as a detector, and you can see the entire image at a time. Whereas in confocal, because of the pinhole, what happens, you see the out of focus light is getting restricted here, and only the light which is coming from the focal plane, it will reach to the detector. And here the PMT, that is photomultiplier tubes, are used as a detector. And in confocal microscopy, the image, the entire image will not form simultaneously. It will be the confocal microscope will scan the entire sample, which is known as the raster scanning, and that we'll discuss in the next slides. So it will scan the sample and it will make the image point by point. Okay, I will show you how exactly the image is formed in confocal microscope. So on this slide, you can see the real illumination volume of the wide field and the confocal microscope. So this is, you can see the slide and the objective lens, and here also you can see the slide and the objective lens. But you see in the wide field microscopy, a relatively larger area is illuminated, whereas in confocal microscopy, a tiny spot will illuminate because of the laser light source. So another major difference between the wide field and the confocal microscope is that in the wide field microscope we use metal halide lamp or the mercury arc lamp as a light source, which is a basically which provides all different wavelengths, and then using the filter we choose the specific wavelength. Whereas in confocal microscope, lasers are used as a light source. So what advantage lasers offers? So first thing, lasers provides a coherent light source. That also provides the collimated light source, and that also provides the high intensity light source. And because of that, you can actually illuminate only one point of the sample compared to the entire big area of your sample. So you can see here the imaging volume is represented for the wide field microscope, and this is for the confocal microscope. So in the confocal microscope, at a time, only this cone of light illuminates the sample, and only this particular point will get illuminated compared to the wide field microscope. So here is another graphics to explain you the light path of the confocal microscope. So how the light path? So generally the light path of the confocal microscope is same as the epifluorescence, any epifluorescence microscope. As you can see, this is the lasers. So basically that is a light source. From light source, one specific wavelength will pass through this beam splitter, and this is basically the filter of specific wavelength. So that light will fall onto the sample, it will illuminate the sample, and the light which is emitted from the sample goes back to the objective, and it will pass through this dichroic mirror and the emission filter, and there is a pinhole here which blocks the out of focus light, and only the light which is in focus will fall onto the detector. So detector is the photomultiplier tube. Okay, so see this most important thing that you should understand here is pinhole is in the emission light path. It is not in the excitation light path. The excitation light path is same as the wide field microscope except the light source, but this pinhole is placed in the emission light path so that it will block the light from the out of focus molecules. So what does the pinhole do? So as we just discussed, pinhole removes the out of focus light. It is adjustable as its size depends on the objective lens used and the wavelength of the light. Then pinhole size is measured in Array units. Okay, so pinhole is just kind of a closed circular ring. Okay, and the diameter of that ring is adjustable. So you can keep it open, and it is measured in the Array units. You can keep it open for one Array unit, or you can keep it open for two Array unit, or any point in between. Okay, so ideally the Array unit of one is ideally preferred because that only collects the in focus light. I will explain you more about these Array units and the pinhole. So in fluorescence microscopy, there is far less in focus light than out of focus light. This is very important statement. See, in fluorescence microscopy, there is far less in focus light than out of focus light. So when you are imaging a sample, okay, your objective lens is collecting the light from the molecules which are in focus. But compared to that light, there is lot of stray light coming from the molecules which are out of focus, either from above the focal plane or the below the focal plane. So for confocal microscopy, a more intense light source is needed, and that's why lasers are used, and more sensitive detectors are needed. So generally the photomultiplier tubes or the EMCCD cameras are used for confocal microscopy. So let me show you how to decide the size of the pinhole and what happens when you change the size of the pinhole. So ideally, the one Array unit size of the pinhole is considered standard, but still your manufacturers of the microscope and the software allows you to adjust the size of the pinhole. You can keep it to 2 Array unit, you can keep it to 0.5 Array unit as well. So sometimes depending on the applications, you have to decide, you have to change the size of the pinhole and to make it perfectly complement with your sample. So see what happens. So this you are seeing the emitted light from the sample. Here you are seeing the emitted light from the sample, and the red ring represents the diameter of the array ring. So here the pinhole is widely open. Okay, so it is greater than 2 array unit. It is too big, and because of that it will lose the confocality. Now why it will lose the confocality? So that you can see here. So consider that this is the completely open pinhole, and on this side of the slide you can see completely means almost closed the almost closed pinhole, which is less than 1 array unit. Now the pinhole is open for greater than 2 array units, so it is almost completely open. And this graph is very important here. So there are 3 lines you can see. The green line represents the signal intensity. Okay, let me explain what is there on the x and y axis. So on this axis, this is y axis on the left side, it is the resolution. The y axis on the right side is the intensity, fluorescence intensity basically, and x axis represents the pinhole diameter in array units. So you can see this is 1 array unit, this is 2 array unit, the size of the pinhole. So what happens? So currently you are seeing this red line here because the pinhole is open more than 2 array units. So this red line is here. So at this position, if you open your pinhole to this size, that is greater than 2 array units, then what will happen? You will collect the maximum fluorescence intensity from the sample. Okay, At this pinhole, the sample will be bright, and you will collect the maximum fluorescence intensity from the sample. So as I mentioned that on the y-axis represents the resolution. So higher numbers represents poor resolution, lower number represents better resolution. So what happens when you are keeping the pinhole completely open at this position? So you are collecting the maximum intensity fluorescence intensity of the sample, but you are compromising on the X, Y, and Z resolution. So this dotted line represents the XY resolution and the continuous black line represents the Z resolution. Okay. So at this position, if you keep the pinhole open for more than 2 array units, you will collect the maximum light from the sample, but you will compromise the X, Y, and Z resolution. What will happen in extremely opposite condition? That is on this side of the slide. So you are keeping the pinhole almost closed, less than 1 array unit, which is not recommended, and it will lead to signal loss. Why? So see, now you are putting your pinhole here, which is lesser than 1 array unit. Okay, so you are only collecting partial fluorescence intensity from the sample. So let's say if you are imaging some samples with low fluorescence, then you may not even able to see if you close the pinhole for less than 1 array unit. Okay, so just to make the sample visible, you may need to open the pinhole little bit so that yeah, sufficient fluorescence intensity is collected and you can see your structures. Okay, but if you close the pinhole to less than 1 array unit, it will give very poor fluorescence intensity. Also, you will get better X, Y, and Z resolution. Okay, if you keep the array unit lesser than 1 array unit, but maybe you will not collect the enough light from the sample. What will happen if you keep the pinhole equivalent to 2 array unit? So here you can see you are collecting maximum light from the sample. So sample will be bright enough, but you will compromise on the X, Y, and Z resolution if you keep the pinhole to 2 array units. Okay, but this is the ideal condition of keeping the pinhole at 1 array unit, and this will provide the perfect confocality. So here, see, at 1 array unit, what happens? You are collecting the maximum intensity from the sample also, and you are collecting you are getting the best Z resolution as well as the relatively good XY resolution. Okay, the XY resolution is not the best, but you are not compromising significantly on the XY resolution, but you are achieving better with the Z resolution. Okay, so that's why 1 array unit condition of the pinhole is generally considered as an ideal condition. Ideally, you should start imaging your sample with the 1 array unit, but later by looking at the actual image, you may adjust it to maybe 0.75 or 1.5 array unit after looking at the sample. Let's say if you are keeping the array pinhole open for 1 array unit, and let's say you are not able to see the fluorescence intensity significantly. Okay, you are not able to see the fluorescence, then you need to increase the pinhole to 1.5 array unit so that you can see your samples. Okay, but if sample is bright enough, and you may work with the 1 array unit or even with 0.75 array units. Okay, in that case, accordingly, you will get betterment in the XY and Z resolution. So always with each sample, and by putting the real sample on the slide and imaging or focusing that sample, you have to decide the array unit for your confocal microscopy experiments. And as per the requirement, you should increase or decrease the size of the pinhole. But start with 1 array unit. Okay, that's the ideal condition. Now here is the field of view comparison between the wide field and confocal microscope. So under the wide field microscope or the epifluorescence microscope, you can see this entire field all together, right? But in the confocal microscope, you cannot see this entire field all together, but instead you will see a small point like illumination. So just a small point at the center of this field is getting illuminated, and you will see that with that point how much fluorescence it has. Okay, so and why this is happening? Because of the laser light source. Okay, because lasers are the coherent light source. You can illuminate one particular point because of the coherent and collimated light source, and that's why you illuminate only this specific point and you image that point. Okay, so how the confocal microscope can generate the entire image of the specimen like this. Okay, so we'll see it here. Point scanning of the image. So how the image is recorded in the confocal microscope. So let's say this is your cell and you want to image this cell under the confocal microscope. So confocal microscope will put a virtual grid over of your sample. A virtual grid means it will divide your sample into so many small boxes. Each cell in the grid is called a pixel. So each square in this grid is known as one pixel. You can decide how many pixels there are, so that you can decide whether how many pixels you want for one imaging. And the confocal scans across the image pixel by pixel. So what will happen? First, it will illuminate this small square and collect how many photons comes from this area. So here the cell is not there, so obviously this field will be dark, so it will give rise to less number of photon. Then it will move to this point. It will again illuminate this point and collect the photons from this area and see how much bright it is. Okay, so depending on the number of photons coming from this particular square, it will convert it into the digital signal. So that way the confocal microscope will scan this image point by point, pixel by pixel. Okay, and it will record the number of photons coming from each of these point, and that will lead to the formation of the digital image. This scanning makes confocal microscopy slower than the wide field microscopy. So confocal microscopy is slower than the wide field microscope, because in wide field microscope you illuminate the entire cell altogether, you can get the image of the cell at the same time. Whereas in confocal, it will scan your cell like this. First it will scan all these 5 boxes, then it will scan these 5 boxes, then it will scan these 5 boxes, then these 5 boxes, and then these 5 boxes. So it will take little long time to form one image. Okay, and point scanning can cause some fluorescent dye to photo bleach. So that is one of the disadvantages. Sometimes because of this point scanning, some of the fluorescent dyes photo bleach faster. And need to use the dyes which are bright. So generally for confocal microscopy, you need the dyes which are bright, because otherwise, because you are illuminating just a one point on your sample. So if your dye is weak, then you cannot see fluorescence in the confocal microscope. So now I'll show you one video from YouTube for almost like 5 minutes to understand this concept of how the confocal microscope forms the image. Okay, so let me open this YouTube video. Okay, so this video is from the Bayer College of Medicine by Jason Kirk, who is the core director of the optical imaging and vital microscopy core at the Baylor College of Medicine. So as such, this entire video is very useful to understand the fundamentals of confocal microscopy, but I will take you specifically to one time point to show you how the image is formed in the confocal microscope. So I'll play this movie from here for next 5 minutes, and you will see how the image is formed in confocal microscope. So this is the field of view of a wide field microscope. And now you will see the field of view of the confocal microscope. It's a point kind of source. Okay. You can see that the confocal microscope is scanning the image in X and Y. Okay, so you can see that entire illumination field moves from top to bottom. So how the illumination takes place? So it puts a virtual grid on your sample, and one point gets illuminated first, and PMT will record the number of photons coming from each of this point, and then this is known as the raster scanning of the image. So your entire image will be scanned point by point, line by line, like this. It happens really very fast. And how it scans the entire field? By putting the XY galvanometer mirrors, so that the entire light source is not moving. Only using the mirror, you can move the light source to scan your sample in X and Y. So this is how your confocal microscope scans your sample and forms the image. Now, how the signal is detected? So, as the confocal microscope illuminates each point and record the photons coming from each point at a time, you cannot use the camera, because camera have the CCD chip, which actually which can form the image of the entire field. But here you have to collect the photons from individual point. So photomultiplier tubes are used, and those photomultiplier tubes have this kind of a mechanism to amplify the number of electrons. So initially, this is known as the input electrons. So only a few electrons are hit the first diode, and then it amplifies couple of time before reaching to the anode, that is the detector. So this is the role of photomultiplier tube to amplify your electronic signal. Another important concept here, generally in the confocal microscope, you will see that the software has a button to increase the gain. Okay, so what is the gain in confocal microscope? So gain is nothing but increasing the voltage of this photomultiplier tube. So when you are keeping the gain low, at that time, a low voltage current is applied to this photomultiplier tube, and that's why the amplification will be lower. But when you increase the gain, at that time the high voltage current is applied for this photomultiplier tube, and because of high voltage, the amplification of the electrons will be more. So when you are imaging the samples which are not bright enough, at that time you should use high gain, so that you can amplify the electrons to a greater extent. But let's say if you are imaging a sample which is already bright, then you do not need the high gain. You can even image with the low gain. So gain is basically the adjusting the voltage of the photomultiplier tube. And this is how point by point your microscope will image the sample, and it will provide the electronic signal to the PMT, and PMT will convert that electrical signal into digital image. So this is analog to digital converter, so which converts your electronic signal into digital image. So this is how the image is formed under the confocal microscope. Okay, so now you have seen how the image is formed in the confocal microscope, and here again a brief introduction about the photomultiplier tube as a detector. So generally in the confocal microscope, photomultiplier tube is used as a detector compared to other microscopes. In wide field microscope, the CCD camera are used, because in wide field illumination, the entire cell gets illuminated at a time, and so that you have to collect the information from so many pixels simultaneously. Whereas in case of confocal, uh, the image is found by scanning the sample in point by point manner or the pixel by pixel manner, and that's why at a time you just have to record the electrons coming or photons coming from the one particular point on the sample. So that's why the photomultiplier tube is used. So advantages of the confocal microscopy. So here I have shown some of the specific conditions in which you will prefer confocal microscopy over the wide field imaging. So let's say if you are imaging a thick sample like tissue sections. So if your tissue sections are 30 to 5 micrometer, then you should use less than 20x objective. If your tissue sections are 10 micrometer in thickness, you should go for greater than 40x or even oil immersion objective lens. If you want to image whole mounts, which are 100 to 150 micrometer thick, then you should go for the confocal microscope. But this is not a good technique for imaging thin samples or the thin tissue sections. Okay. Then confocal is unavoidable in case if you want to go for the photo bleaching based experiments such as FREP, fluorescence recovery after photo bleaching, that we studied in one of the previous class. So for doing FREP analysis, because there you have to make the specific ROI to bleach one particular area of your cell. So that thing is not possible with the wide field illumination, because with wide field you cannot focus the light to any small part of your sample, whereas using laser you can focus that light specifically on one particular part of your sample, and you can bleach specifically. So for any photo bleaching based experiments, you should use confocal microscope. And also for any photo activation, photo convertible fluorescent proteins and photo switchable fluorescent proteins, for those kind of photo conversion phenomenon also you should use the confocal microscope, and this kind of ROI selection is not possible with the wide field illumination. And also in some of the samples where there is a heavy overlap between the emission wavelength, let's say you are imaging GFP and YFP in the same sample, then you should go for confocal, because in confocal you can spectrally separate these emissions, which is not possible in the wide field illumination. So coming to the disadvantages of the confocal microscopy, in which condition you should not use confocal microscopy. As I mentioned that most of the application, or 90% of the time, you may see what the details of your samples using the wide field illumination. In that case, you will not go for the confocal microscope. So some of the disadvantages of confocal microscopy are listed here. The first thing is the slow acquisition, because as I mentioned that it will scan your image pixel by pixel, so it will take long time to scan the entire image compared to the wide field microscopy. And longer scans means more exposure. Okay, so if you want to image very sensitive sample, okay, live cells or anything, then if your fluorophore is weak with low quantum efficiency reporters, then you should not use confocal, because of the lasers. It will really create a huge photo damage if you are imaging a live samples. And if your fluorophore is weak, then you cannot even visualize it clearly using the confocal microscope. And if your fluorophore is bleaching faster or it is creating a lot of photo damage, you should avoid using confocal microscope. And little or no benefit for the thin specimens. If the ultra thin cryo section less than 10 micrometer cryo sections are there, you should not use confocal, because you will not get any added advantage. Okay, and again, longer scans means more exposure, and that will create more phototoxicity and photo bleaching in your sample. So in summary, if you are imaging the thick samples like thick tissue samples, or maybe the entire humans eukaryotic cells, then you should go for the confocal microscopy. So now coming to the little advanced version of the confocal microscopy, which is known as the spinning disk confocal microscope. And as we know that this confocal microscopy is generally slower imaging technique compared to the wide field imaging because it scans point by point, right? So how we can increase the speed of the scanning? So spinning disc confocal microscope was designed in which you can see in wide field you are illuminating the entire sample, whereas in laser scanning confocal microscope you illuminate only one particular point and take the image at a time. Whereas in spinning disc confocal microscope, you can see that you are illuminating multiple point at a time and you are detecting the illumination from these multiple points. So basically by illuminating multiple points instead of single point, you can increase the speed of confocal imaging. So how this spinning disc confocal microscope work? So initially this technology was designed by the Yokogawa spinning disc. So this disc that you are seeing which multiple holes, that is known as the Yokogawa disc because it was discovered by the scientist named Yokogawa, a Japanese scientist. Okay, so how this works? So see, this ring rotates very fast, and because of that what happens, let's say this is your specimen, it will illuminate multiple points simultaneously instead of a single point in the regular confocal microscope. Okay, so see how actually the light path works. So here is your laser. Okay, this is collimated laser illumination. This is your laser beam. You are putting this Yokogawa disc in the laser light path. So this beam is splitted into multiple points, and then you are putting some appropriate filters and dichroic mirrors in the light path, just like the filter cube that we use in the fluorescence microscope. And then this disc will illuminate some specific points on the sample. This is your specimen. So see, the entire specimen is not illuminated at a time, but instead a few points are illuminated. And the few points will emit the fluorescence light that will be detected back by this another disc. This is a pinhole actually. Okay, this is a disc for the pinhole. Okay, so that disk, the emitted light will pass from this pinhole disk and it will be captured on the camera. Now see, here it's a multi-point detection, so you cannot use the photomultiplier tube. So if you are using the spinning disc confocal microscope, you have to go for the CCD camera because here the multi-point illumination is there. So multiple points cannot be detected by photomultiplier tubes, and you need a CCD camera or EMCCD or SCMOS camera. Okay. Now, one limitation of this spinning disc confocal is that this pinhole size is not adjustable because this is a disc, right? It is not just one pinhole that you can increase or decrease the size of that single pinhole. So here it's a pinhole disc, and that's why the size of this pinhole is fixed at 1 array unit. So here are some differences between the point scanning confocal microscopy and the spinning disc confocal microscopy. So this is known as a laser scanning confocal microscopy, in which the image is scanned in this manner, whereas this is a spinning disc confocal microscopy, where because of this disc, multiple points are illuminated and you will collect the information from multiple points, so your imaging will be faster. So this laser scanning confocal microscopy can provide high confocality and better image quality compared to the fast acquisition by the spinning disc confocal. So image quality, if you see, it is better by the laser scanning confocal microscope compared to the spinning disc confocal. But let's say for some you are imaging live samples and speed, you are really looking for the high speed imaging, then you should go for spinning disc by compromising little bit on the resolution side. Okay. So laser scanning confocal provides high resolution, whereas this is low dose excitation or offers low dose excitation and low phototoxicity. So as previously we discussed that this laser scanning confocal microscope is very detrimental for the live cells because it may lead to photo bleaching and photo damage, okay, because of the high laser powers. Whereas here it's a low dose excitation because the laser beam is splitted into into multiple points, so the dose of the laser power is shared, okay, and because of that it creates low phototoxicity. This provides increased depth. So in Z, it provides its Z depth that is covered by the point scanning is higher than the spinning disk, and here the frame rate is high, so you can go for fast imaging. This is good for fixed tissue and this is good for live cell imaging. Why it is good for live cell imaging? Because of the low phototoxicity and fast imaging speed. Whereas this is good for fixed cell because with intense laser power, it will there is no issue of photo damage if the samples are fixed. Okay, and that's why this laser scanning confocal is good for fixed tissue. Then frame rate of imaging. So this can provide 1 fps, means 1 frame per second, whereas this can give 30 to 1000 frames per second. Then here the detection is photomultiplier tube based, whereas here the detection is camera based because it's a multi-point illumination system. So you need a camera instead of a photomultiplier tube. And here the amplification signal is amplified, and here the pinhole size is fixed. Okay, and the pixel size is also fixed because of the Yokogawa discs. Okay. So coming to the last slide, and here you can see for what applications the confocal microscopy is used for. So confocal microscopy can be used for localization and co-localization of proteins if you want to do multicolor imaging. Then imaging two or more fluorescent stains, the same thing like multicolor imaging in 3-dimensional, 4-dimensional, 5-dimensional. 3D visualization of staining, quantification of protein expression, and acquisition of high quality images for presentation. So this last point is actually very important. So generally for most of the applications, what people do for your regular imaging, you may use your wide field microscope, but just to produce one high resolution, good quality image for your publication or for your presentations, you will collect one good image on the confocal system so that it will be more precise, or I would say you can see here in the very beginning I showed you. Let's say you just have to show some representative image, then it is better you take that image using a confocal microscope so you can get crispy image. Okay, whereas in wide field you will get this kind of image because of the little blur. This is a little blurry image because of the out of focus light. So just to make one representative image, you go to the confocal system and get one image so that you can show it in your presentations or in your publications. But let's say for rest of the applications you may do imaging with a wide field microscope. Okay, so that's all about confocal microscope and for today's class. And our next class will be on super resolution microscopy, in which we will discuss three types of super resolution imaging techniques. First one is SEAM, that is structured illumination microscopy. Then STED, that stimulated emission depletion microscopy, and the STORM microscopy, that is stochastic optical reconstruction microscopy. Thank you and see you in the next class.